to allow it to keep on getting ATP when the situation is not otherwise working optimally, right? Not working to the best. So we do say this is anaerobic because glycolysis, right, the breakdown of glucose, is done without, right, again, without oxygen. And this is very key. This is advantageous from the standpoint if I don't need to get oxygen there, again, I can still get ATP. So the obvious is not that you stop breathing. Why wouldn't I have oxygen, right? It isn't that you stop breathing. It's delivery, right? It's delivering the oxygen to the muscle cells out here. Because the only place oxygen comes in is the lungs, right? And so it's got to get to all these active muscles, which means it's got to cooperate, right? Lungs are cooperating with the cardiovascular system. The heart is pumping at a certain rate, and the blood is moving, right? And then the gases have to diffuse. Okay, well, you could be so energetic needy that you couldn't get enough oxygen to do what we're going to talk about next, aerobic, and so you therefore would do anaerobic, right? So you still get ATP in the absence of oxygen, but you don't get very much. Most of your energy, because this has got a lot of potential energy in it, right? Calories. Most of the energy is still tied up in this molecule we call pyruvate, right? But ATP can be made a little bit, right? If oxygen's around, you will go on to aerobic, which we talk about next, but no oxygen, right? It's anaerobic. And so you can keep getting ATP, again, the point of the slide is you can keep getting ATP even when you can't deliver oxygen, okay? So lactic acid is the outcome of anaerobic. It's either, no, we don't do fermentation, but that's other organisms do. We just call it glycolysis, right? And this, of course, contributes to fatigue because fatigue is running out of ATP. And you are going to run out of ATP pretty quickly in this situation. You're only going to be able to continue doing glycolysis for maybe 2 or 3 minutes. It's just, you can't continue it. Just like you're going to run out of creatine phosphate. You're going to run out of, right, energy, ATP, adequate energy, and you're going to decline. So again, fatigue, we really say we are consuming or using more ATP than we can produce. And that's going to happen here, right? Because that's not efficient. I don't get much ATP. Most of the energy is still tied up here, all right? So I say again that the take-home message is here, still works in a strenuous or intense activity when you need the energy, but you can't deliver enough oxygen. And don't be confused by the more. It needs additional energy. I need energy, but I can't get oxygen. All right? So, whoops. Here's a thought, right, when we want to put this together and think about the big picture. Again, you're in a situation that ATP is being used faster than it can be produced, right, in intense exercise. So you can't efficiently make enough ATP to keep the muscle moving, but you can still make ATP. You'll convert the creatine phosphate back to creatine, right? You'll get ATP, you get about 15 seconds. That may be enough. Once you deplete creatine phosphate, oxygen is in short supply, you'll do anaerobic, right? Glucose, from either blood glucose or glycogen stores in the muscle, will be broken down. That's what catabolize means, right? Broken down. And you get about, maybe 2 more minutes of ATP from that. Just, these are limited, but they keep you going, right? Because we said if we run out of ATP, we cannot contract and we cannot relax. So we stop in our tracks. It's called physiological contraction, all right? So that's the idea that you can keep getting some ATP, even when there's limitations, okay? So this is the standard. We are really evolved to do this, right? And in that muscle, we're going to have the ability to produce even more mitochondria. Mitochondria have their own DNA, they divide, and when you put enough stress on a cell, that will trigger the division of the mitochondria. We call that the training effect. Just like that stress will trigger the development of more actin or production of more actin and myosin. Because we realize the more actin and myosin we have in a muscle, the more force we can generate, the more we can do. That all comes about from training, doesn't it? Weightlifting and aerobic training. All right? So, very efficient. It takes longer to do this because these are like 40 chemical changes occurring in this process we call aerobic respiration. It's important in the cartoon, shows you this idea, mitochondria, right? This is the key place. And in this terms, we can use blood glucose, we can use pyruvic acid, so the pyruvate that we looked at there, we can shunt back into, it gets moved into the mitochondria. We can use fatty acids and we can use amino acids that aren't shown here, right? The beauty of your biochemistry is that it's actually adaptable to all your macronutrients, your big nutrients, your lipids, your carbohydrates, right, and your proteins. So I can use a variety of different starting molecules to do this. So that gives me an advantage, right? I'm not locked or dependent on one specific. And as the simple cartoon shows you make a lot of ATP, a lot more. All right? And so this is a full breakdown, really, if you think about it, of the release of as much energy as possible to ATP. And then really the rest goes to heat, because that's the second law of thermodynamics. But that's okay. We get a lot of ATP. We can keep those muscles working. All right? So the idea is we'd really like to do this. Right? That's really the optimal, as we said. So here again, oxygen is required. So I say, right, I just remind you, the lungs bind oxygen. Oxygen doesn't stay in solution. Your blood is a solution. It's not very soluble in fluids. It really likes to be in a gaseous state. So you've evolved this lovely protein, hemoglobin, that there's billions of molecules of hemoglobin in a single red cell. There's probably 100 million, but there's a lot. And they pick up that oxygen in your lungs, in the alveoli, and they bind to this protein hemoglobin. It turns bright red when it binds. Beautiful color, right? And it's transported to the cells, as I said, and from the cells it diffuses, right? Because we know the laws of molecular movement. You guys learned that because it's high concentration of oxygen in your systemic capillaries. It's lower in your cells, so oxygen moves into the cells. And here the cells being muscle cells, right? We got one more advantage in a muscle that we don't see other places. And this is another molecule I told you in your reviews you should know. And we've got we've got myoglobin. Myoglobin is very similar to hemoglobin. Furthermore, it works like hemoglobin does. It actually binds onto that, holds onto that oxygen, so it can store oxygen. Because that hemoglobin, it just unloads it, it moves off of the hemoglobin, it moves in, and the hemoglobin moves on, back to the body, and hemoglobin is going to continue moving between the different parts of the body all through your life, carrying oxygen. Right? And it carries some other gases too, but oxygen is what we're focusing on here. So here's the scenario. So that hemoglobin in the capillaries releases the oxygen moves into the muscle cell, right? And it would normally, in another cell, be used right away or gone, right? And here in muscles, though, we have a special protein that can keep it. It's not gone, so keep it, storage. Now, this presence of myoglobin actually changes the appearance of the muscle. So we're going to talk about this in just a second here, but you've surely heard about light meat and dark meat, right? So dark meat is dark because of the presence of this molecule. So the idea, again, here would be that if a muscle was more active, right, needing more ATP, it should have more myoglobin. Would that make sense to you guys? Right? So myoglobin, again, so it can hold on to oxygen, which means it can do more efficient energy conversions, right? Because these guys all have really a lot of potential energy, and we're trying to get it to ATP. So that is evident in the actual color of the muscle. Yeah, Kim?
Does myoglobin have iron in it? Like
Yeah.
Okay.
All right. So I just remind you in italics that the issue here is that a muscle contracting can actually intermittently stop blood flow. Just for a very brief period of time, but blocks it because it's pushing against it as it shortens. And so having a storage molecule is really important here in keeping this muscle going. All right? So now we all face this. I mean, maybe you've never faced this, but if you haven't, I always say to people, look, you should push yourselves. You're outstanding machines, and you should figure out your limitations. Because only by figuring out your limitations can you overcome them. That's my thoughts. And I'm not a drill sergeant. I'm just saying that really we are fine, well-designed machines. Let's make them work. So it's good to push yourself, right? Now you're going to do a little damage, but again, if you're young, you're going to repair that damage, right? So, but here, this is the idea. So if you've never experienced this, then this might not be easy to relate to. But I have. I know there's plenty of times. I mean, and I'm not talking about the psychological fatigue that you have when you leave this room and you're like, I just said a lot of big words and I've got to figure out what they all mean or something like that. Not that kind of fatigue. I'm talking about, again, physiological fatigue. All right. So I don't know that they do this anymore. But when I was growing up, everybody took physical education, right? And we did wind sprints, right? And we did 50-yard dashes and 100-yard dashes, and it was all part of what we called the presidential fitness. Though I'm not sure that all the presidents we've ever had are in great shape. In fact, I wouldn't argue that they are, right? But that was what it was called. I don't know what they call it anymore because now my son's in band, so he gets an excuse, right, for marching band. He doesn't even have to take PE. So, but yeah. So I am very familiar with the idea of running up and down because some masochistic, well actually sadistic. It wasn't masochistic. Sadistic gym teacher says, hey, you guys need to do that, right? You guys probably get it with your coach, right? Gets that pushing you guys to the point of, and you guys, some of you are softball, right? Anybody? Nobody softball? So it's just baseball still? Okay.
Nowadays they have the FitnessGram Pacer test.
Okay, all right. Fitness, they have to. Is there a nice acronym for that? All right. So again, the point being is that you'd push yourself. So you'd be doing intense activity, right? And so I say what would happen is, and we'll talk about this in the next slide, is you'd get done doing that activity and you'd be doubled over, breathing heavily. And that's what we call oxygen debt. You have incurred an oxygen debt because you pushed the muscles so hard. They've used up their myoglobin, they've used up their storage of creatine phosphate, and they need to be paid back in amounts of oxygen. All right? So this slide is really saying, look, fatigue, just simply stated, is you're using your energy molecule faster than you can make it. Faster than you can make it by creatine phosphate, faster than you can make it by glycolysis, faster than you can make it certainly by aerobic respiration. So you're switching, right, between these options. And I say the consequences are that you get this molecule, lactic acid builds up, all right? And that affects the environment of the cell. And you have ion imbalances that occur because you've got all this electrical excitation occurring. Calcium has to be available. Sodiums and potassiums are being altered in where they are in the cell. So again, what you want to think about here is just there's a limitation to the ions you have. And you are experiencing an imbalance, right? And you tend to do some muscle damage because of this situation as well. So you do have to repair that. So, all right? So I say here again, the point of this slide is that in fatigue, contractions will weaken because ATP levels are dropping. And calcium, as well as the other ions, are just not, there's not a limitless supply. So this is a reality check, if you will. Again, the limitations of the machine. Okay? So oxygen debt is already mentioned in an intense exercise, especially intense, because in aerobic exercise, we're implying by the name that you are getting, that you're pacing yourself enough to deliver the oxygen to optimize your ATP production and be efficient. But in intense exercise, that would be compromised. All right? So I say, when you get done, because you can't sustain this, right? This cannot be continued, again, because of the inefficiency of glycolysis and how little creatine phosphate can store in energy. So you finish and you have to restore everything. You convert that lactic acid because it's still got a lot of potential energy in it, and you're basically resetting the muscle. So this is advantageous if you're trying to, again, get away from something dangerous or deadly, and you just need enough time. So this would be an evolutionary advantage to us as a species, as it is to other species. In fact, if you look at predators in general, and humans are pretty good predators, a lot of them, if they don't capture the prey in the first mile of the chase, or several minutes, they actually stop. Because there's limitations to the ability to continue to efficiently and get enough energy. So that's why many predators are ambush predators, right? They lie in wait. And then they have reserved their energy to the point for the strike and the movement after the prey. But if they don't make it, again, if you're talking about wolves in the first mile, alligators is like the first, 50 yards or less, right? If they don't get it, then they're going to have to wait for resetting their muscle. So their success lies on the ability to do that. So that's what I'm saying. As a prey item, because we are both predators and prey, right, it's advantageous that you would be able to overcome, right, be much more efficient. And all you have to do is sustain it a little bit longer. That's really the reality, isn't it? So this isn't just an accidental coincidence or happy coincidence for us. This is evolution in process right here, right? Because those that live go on to have offspring, right? Those who die are selected against. So it's very interesting, and please approach the information as such. It's not something, again, that you choose to do, right? It's something in our genes, so it's subject to selection. Yeah, Kim?
They had this really interesting thing where they had Kenyans, an indigenous group in Kenya.
Mm-hmm.
They would run 20 miles, and they would just run a deer out until it got tired, and then they would just sit down on its knees and just stab it with a spear. And
yeah.
That's how they would catch their deer. And
Yeah, there is a genetic advantage to a number of different people.
Dwarves are sprinters.
And I'm not sure I buy that.
That's what I mean. Anytime I'm like running with somebody and I start falling behind, I'm like, we dwarves are sprinters.
Yep, I gotcha. So, do we still not? Oh, I turned the sound down a little bit here. My first class. Yes. All right. Hang on. Hopefully Jade will come back. Devin's not here today. Amanda's here. Alex is here.
Oh, that's so cool for the Cahoot. They have
David, Tanner, Mariah, Kale, Alexia. Yes.
I'm just putting this aside.
Landry. We got 5. Is that it? Yeah, because she's not here. So we're all. You guys are all logged in, right? Okay. Jane has stepped out. 8 questions. Let's see. Hopefully, some of my wording might be a little bit, because I don't know if I edited these from the last 2 holes. Initiates a muscle contraction. What's going to be the first thing that you're going to want to do? Yay! Yes, please. The release of acetylcholine is where we're going to start here. We're going to back it up in go a little bit further back to talk about some events that precede this. But for us, we're starting with this acetylcholine. We're getting this idea that the nerve is telling, right, the muscle what to do. So we're not going to get to the binding of calcium to troponin for a while. So again, you can't contract if you don't have acetylcholine. That's why I said we have to back it all the way up. All right? What's the protein that binds calcium and allows cross-bridges to form? So simple vocab. Yeah, it's troponin. Troponin has the actual calcium binding sites. So again, these are questions I would truly see on my test. I mean, so please, if you're not getting them right, then you know where you need to focus some attention. What is the unit of contraction called? This one probably I won't say on the test, but who's actually responsible? Where does that contraction begin? We often use unit to get to the small ends. Yeah, sarcomere is definitely. What happens to the sarcolemma in response to that acetylcholine? Yep, it is a sodium channel. So it's step 1, step 2. What's true for relaxation? So what would we do when we're relaxing? Yeah, a little bit of division, right? No, if you keep releasing acetylcholine, we're just going to keep on contracting, right? This is the key, again, to tell the muscle what to do. So that's not going to work. The calcium's being pumped back, right? We're getting rid of the ingredients, if you will. Got to have calcium so that we can, right, move tropomyosin, which means that actinomycin can bind. So if I pump it back in, troponin and tropomyosin go back. Not necessary for contraction. Which one's not necessary to allow contraction? Yeah, lactic acid is not necessary, right? Lactic acid is what's going to form, right, if we have to go anaerobic, do glycolysis, but that's not necessary. Calcium you got to have. ATP you got to have, right? Acetylcholine you have to have, right? So that's, again, in ATP, that's our only source of energy. So refers to an increase in force. Oh, we haven't covered this yet, but that was in your pre-chapter. An increase in force with an increase in the number of impulses or action potentials, if you like that word better. So this is a preview. Sorry about that, but certainly opens a conversation. Okay. Just to give you a little bit of a hint, and this is both the face-to-face class and the online class. I had on the pre-chapter quiz a term for an increased force due to the increase in number of motor units. And only 19% of your class got that correct, and only 14% of their class got that correct. That is recruitment, right? If you increase the number of motor units, you are recruiting a friend, right? You're getting somebody else to help you. If you increase a force with an increased number of action potentials, we actually refer to that as summation. And I did say on the review, no, let me see which number it is here. Yeah, somewhere in there.
Action potentials are interchangeable with impulses?
Yes, they are. And I have it somewhere here, and I can't locate it readily on the number list here, but yes, you have to know, right, what the idea of summation and recruitment are. And summation, again, recruitment, I'm getting more motor units. All right, I'm doing exactly what it says. Summation, and I'm going to show you this, so hopefully it'll stick in your head what it is. I'm simply stimulating the same motor unit more often, okay? And I'm going to get more force, all right? Because I said following an increase in force. Force is what I'm after always. Remember that, right? What I'm telling you, there's several ways to get it. And I give you, again, recruitment is an obvious, I will get more motor units to a point, right? Because there is a limited number of motor units in any given muscle that you guys are studying and naming on your practical. So quadriceps, right, this group here, and we look at the rectus femoris specifically, or the vastus, right? Big muscles, lots of motor units, right? Potential there. So I could get a lot of force if I needed it, which is great because I got to move this mass through space, okay? Space and time. So, right, understood. And when I want to do, some movements take more effort than others. So again, effort being, I have to generate force. So summation isn't always so obvious to students. And again, we'll get into it in the PowerPoint. But summation is, well, if I keep sending an action potential more and more frequently, can I get an increase in force? And the answer is yes, within just one motor unit. And I could be doing this in all the motor units, by the way. But yes, I could. And the logical reason I can is because if I keep on stimulating the same motor unit just more often, it's going to keep releasing calcium. So the first time that action potential spreads from this motor end plate here across the sarcolemma and enters that T-tubule, right, remembering the way a contraction works, and the T-tubule changes its polarity, that causes the sarcoplasmic reticulum to open its calcium channels. Well, they only open for a short period, and only some of the calcium leaves. So if I just did one stimulation, I stopped, right, just did something like that, a little bit of calcium would be leaving, but not the whole thing. And then if I relax the muscle, I'm going to pump it back, right? And I start all over again. Well, if I keep stimulating it, well, then more calcium can leave. And calcium wouldn't be pumped back because I didn't relax. I didn't go into a relaxation, did I? So I can keep, what's that?
Is that what makes your muscles?
Well, that's part of the lactic acid that we've talked about there and some of the changes that go with ion imbalances. But in this scenario, right, if I keep sending calcium out, well, then there's more and more opportunity to bind to more and more troponin, which then moves tropomyosin, which means more and more actin and myosin can bind. And the more actin and myosin that can bind, the more force I can get, right? Oh, yeah, there's a limit to this, but please understand, right? If I keep sending more action potential, then I can continue to release calcium to a point, and I can get a stronger, more forceful contraction. It's very physical when we think about these, but you have to think down on the microscopic level. All right, so that's the idea of summation. We'll look at this here in a second. Last question. Which of the following describes an active muscle? So if you're active, again, not what you are right now, probably, right? So what's going to be a really good? Yeah, and you guys get that. I said that you're using the ATP faster than you can make it. You're active. You're not going to be able, you're not going to have a surplus to do this. And you're not going to have a surplus to do this, right? That's really the point. And you're certainly not going to do that because that's, as I say to students, that's your, right, those are events like this are what you do when you're in the feast state of physiology. You got plenty of energy, your needs aren't great, and you want to store it up for a time when you won't have a lot of energy, right? So that's the idea, right? So, right. So, coming back again, zooming back out and thinking about these muscles as a whole, because we spent a lot of time talking about how they work microscopically, and we have to, down in the sarcomere, you can see that. So look at the whole, we define types of contractions. Now, please understand, this is an introductory book. We're trying to make everything as black and white as possible so you can begin to get the basic rules. I'm not going to, in any way, shape, or form, convince you that it's just as simple as you're either this or that. You are a combination of these events happening. But in order to first get you to understand it, we have to present it like, okay, you have some contractions that are just this or some that are just that. Your movements are complicated. And your needs are complicated, okay? So, isotonic comes in concentric or eccentric. So isotonic, same force. That's what the basic word is trying to say. So, all you do, please, is what you have to do in a contraction. So, once you've generated the force to move this weight, and remember its force is gravity on the mass, you're going to move it. And you're not going to generate any more force. That's why we call it isotonic. So, but the simplest way to think about this is that this is a muscle that shortens, or changes its length, I should say, because it's shortening and lengthening. So it changes its length. So once you've achieved the amount of force, again, coming down to how many actin and myosin can bind, which is due to how much calcium is there, which is due to all the ATP, which is due to acetylcholine. Once I've achieved the necessary force in here to move this, and I'm moving it by flexion, right? I shorten that biceps. Because there's a lot of flexors, but you just studied the biceps brachii. So, right, insertion here in the radial tuberosity. So you're bringing that insertion towards the origin. And that's an isotonic. So if that muscle is changing in its length, we call those isotonic contractions. So we automatically know if you're not changing in length, you're going to be isometric, which means same length. All right? So concentric contractions, right? Force increases, but then it's maintained. That's why it's isotonic. It's maintained while the muscle shortens. You're not going to keep building up force. Why not? Because you might throw that weight right over your head, right? You know, I always say to people, look, you got to meet the need, right? Exactly. If you keep on generating more and more force, you're going to projectile it. So this is an isotonic contraction. You generate the amount of force you need to move any given object, right? And you do it. Okay? And that's an isotonic. So that movement, when it's shortening, is concentric. When it's lengthening, it's eccentric. And honestly, I read here recently that it's problems with your eccentric contractions that cause a lot of damage, because people that move things often too quickly put them down and they tear. They make micro tears in the muscle and the muscle tissues. So when you do weight training, they say count up and count down, and they're not kidding, right? That's you're training the muscle by doing both of these, right? Not just lift it up and then drop it suddenly. That's not good for the muscle, right? So your right, concentric is a shortening, eccentric is a lengthening, right? And so you've got these again, and it gets complicated because, like I said, when you're also extending the arm, you've got some of the triceps being involved. So I said we're really presenting this very, very simply, but that's okay. So I'm just going to ask you for definitions of things and say, all right, the contractions don't need to move, right? So we're seeing this muscle, right? No movement. So again, you can hold. So no flexion, no extension, no abduction, no adduction, right? No rotation. You get the gist, right? So this is isometric because, again, the sarcomere isn't changing ultimately. Now it changes a little bit, ever so slightly, but really the overall effect on the muscle is it's not noticeable. So the actin and myosin are simply staying in a bound state, because if they're not bound, you relax, and this is a contraction. So this is often your posture, right? If we think about what you do on a day-to-day, all-the-time basis, is you keep your head up, you keep your back straight, you hold a position. Those are isometric. You're not moving all over the place as you sit there. You might be moving your head a little bit to see and everything, but for the most part, we spend long periods of time in isometric contractions, right? If you think about it. Okay? So again, you have to increase some force, but the muscle doesn't really change at the angle of the joint. Again, what you see isn't changing, and you're not going to move a load. You're not going to move this weight or move in relationship. Right? And this can also be, in a circumstance where something is too heavy. But again, if we're looking at normal, here's what I do. It's your posture. It's holding a position. Right? Stabilizes a joint. We have all kinds of isometric contractions going all the time, because even when you're moving, like that's why I said your movements are very complicated. All right, we're making them look really, really straightforward, that you do this or that, but the reality is you're doing a complex variety of movements at any given joint. But in order to first understand it, we have to kind of break it down. Okay, so please understand, on the test, I'm going to say, well, isometric is defined as this, isotonic is concentric is this, eccentric is that. You know, I might give you an example like, you know, posture, holding your posture is isometric, but that's the idea. All right? So now, this is our idea of changing force. Okay, and this is nice. We just bring back this picture again to remind you, because you cannot get away from the idea of what's going on microscopically. But, right? So I said here, every muscle fiber that is contacted by one motor neuron, right, is a motor unit. Now, I've talked a lot about this already. So this is just in the book, in the chapter, they formally say, here's what we have because we want to talk about summation and recruitment. All right? So again, you release acetylcholine, every fiber is connected to response, right? If they have acetylcholine receptors, they're going to respond, right? And those receptors are going to cause these gates or channels, sodium channels to open. I've already said this on a previous slide, but again, just reinforcing. Motor units can be small for fine control. They can be large for gross motor control, right? So this is not new information on this slide, hopefully. That's something you all have known or had been exposed to, either from me talking, or I know I mentioned on a previous slide about the motor units, the number of motor units. We have talked about that. So, how do I vary force? And again, that was a question of recruitment and summation. How can I change? Because I live in a world in which I have to change the amount of force. The amount of force to hold a pencil is way different than the amount of force to move that desk, right? So you have to match the task to the muscle. And muscle isn't going to be wasteful. As I said, otherwise you'd be throwing things. You'd have no control over movements if you didn't match the force to, right, the actual mass and its gravity of the object, right? So recruitment, straightforward. Here's a couple of different, right? So here's one motor. So these are, that's a motor unit, and then they color code another motor unit in purple. So I could excite this one, and then I can add that one if I wanted to. And that's just a very small window of all the other potential motor units that could be there. And of course, how many cells this contacts really can vary widely as well. Right? So recruitment, plain and simple, is just you excite additional motor units. And so when you train, that's why I said that's why you get stronger per se, you generate more force, is you can produce more actin and myosin, which you can get, right? You don't get new cells, but you add more and more, right, proteins to the existing cells, and that means you can get more force. So there's a training effect that you can easily see here. But we are limited. Everybody needs to understand that you all have different numbers within a range of motor units in these muscles. And ultimately, the force you generate, the limit of that is going to be due to, is everybody excited? Is everybody on board? Okay, so, and I say here, generally you don't recruit all those units unless you absolutely need it, right? And if you've done this, you know those intense exercises. And it's very energetically, when I say expensive, I'm talking about ATP, right? You need a lot of ATP, and that means you'll use it up fairly quickly. That's what I'm saying there. So generally, we only, again, involve who we need, or what we need, I should say, for the task at hand. All right? So this reminds you that there is a physical aspect, and I've said this all along. You only have so much actin and myosin, you only have so much calcium, you only have so many receptors, right? So there is a limitation. So I did not write any test questions on this, I'll be honest with you guys, but this is something to consider. That what you're after is, right, as much force as you can get, right? Tension, force. And so there's an optimal, right, overlap. And actin and myosin have to overlap, because if they don't make contact, you're not getting any force, right? So here we have this stretched muscle. And actin and myosin can't contact, you can't generate any force. And yeah, that does happen. Okay, so if you're wondering about that, that happens. There's a cardiomyopathy. It's called dilated cardiomyopathy, because cardiac muscle has the same arrangement, where you overstretch through time. Your cardiomyocytes, specifically the sarcomeres of the cardiomyocytes, and you can't get the heart to contract effectively anymore. All right, so there's an optimum because to get us started, we have to bind these two things together. We have to understand that physical relationship. So that's really what they're talking in here, right? So I say here again, if the sarcomeres are stretched, not enough overlap, tension falls. If the sarcomere is shortened too much, you can't generate any more tension, and it will also diminish there. So I say it's just right at about 80 to 120%. Again, I did not write a test question, I'll be honest with you about that. But that is a concept to remind you that this is a very physical property that we're talking about here. It's the arrangement of the proteins. All right? So here's our summation, frequency of motor neuron stimulation. Right? So what we call impulses or action potentials. All right? So I say here again, a single action potential produces a single contraction, right? And we call that a twitch. So this is not the problem with a twitch in the eye or a twitch in a spasm, because those are more spasms. This is just the term we use for this process that happens. And the process is latent contraction, relaxation. So it's just giving, if we can, check the tension generated by a single motor unit, right? And here again, you have time, so very, very quick, really quick. So I say to students, and a lot of my online students who actually answer those questions for credit, they just totally miss the opportunity. I mean, they did pretty well on their answers, but they just missed the opportunity of actually applying what they've learned in this chapter to this picture, because that's what you're supposed to do. So you're supposed to tell me, knowing that here's where acetylcholine is released at time 0, tell me what this means and what you already know. Well, here in the latent period, well, that's where acetylcholine is diffusing across the synapse. It's binding to its acetylcholine receptors, It's opening voltage-gated sodium, ligand-gated sodium channels, It's starting an action potential locally at the motor end plate, there, the motor unit. It's spreading that to across the sarcolemma, depolarizing, voltage-gated sodium channels, spreading it into a T-tubule, okay? It's moving down, depolarizing the sarcolemma of the T-tubule, which causes the depolarization of calcium, voltage-gated calcium channels in the sarcoplasmic reticulum. The sarcoplasmic reticulum is releasing calcium. Calcium's diffusing out into the sarcoplasm. It's binding to troponin. Troponin is shifting its position and causing tropomyosin to move. The binding sites on actin from myosin are now revealed. That's there. Everything I just said happened right there. Period. Because you can't start tension until actin and myosin bind. So a lot of students, like you guys did for me in the Cahoots, wants to start this with calcium. You can't do that, all right? You have to back it all the way up. No, on your test, are you going to do what I just did up here? No, you're not. Because you might not be ready for it yet. You will be eventually, but maybe not right now. But you better understand, that all of these things have to happen in order for us to get actin and myosin together. And if they don't, anywhere along the line, we're going to fail. So contraction is going to begin, and all this is very fast, less than, usually it's about 1 or 2 milliseconds. They've got about 5 here, but usually it's 1 or 2 milliseconds. Milliseconds, thousandth of a second. That's why you can do things so quickly. And that's why it's so hard to understand or apply this, because you're thinking, all this is happening, and it's happening so fast that it's easy to miss. So it's at this point then, when actin and myosin touch, bind, form cross bridges, all those terms that we use for this, that we start to get tension. All right? And if this wasn't isometric, we wouldn't really see a change in tension. We'd see it rise a little bit and then stabilize. If it was concentric or eccentric, we would see this change. Okay, and so here's the period in which actin and myosin are binding. And while they're binding, I always have to have calcium around. I always have to have ATP around, because those cross bridges are forming. Now, I don't actually know if this muscle is shortening or not. I said concentric and eccentric, but I don't actually know that, because that's not measured on this graph, is it? I mean, I'm just going to be honest. I always say to students, look, just tell me what you can see on a graph. Don't make any judgments that are beyond your ability. Always use what's in front of you and say, here's what we know. All right, so this one's a very short, and then it relaxes. So the tension falls. So here again, during this whole time, actin and myosin have to be making contact. ATP has to be around, right? Calcium needs to be available. Everybody clear on that? All right. So then, when you're going to relax the muscle, then you got to start pumping all that calcium back. So you still have to have ATP, right? The actin and myosin come apart and stay apart. That's the key there, stay apart. Okay? And everybody resets. Tropomyosin goes back and covers the sites, right? And so this would be all during this period. And this period could go on. I mean, the time is going to vary clearly for contraction and relaxation. The time is probably not going to vary for this, what we call the latent period. All right? But this is all what we've already talked about. This is not new either. Just like I said, the idea of being able to increase contraction, that motor units have different numbers of fibers and so forth, that's not new for us. All right? But that's what we want to, where we want to be. We want to be looking at a chart like this and thinking, okay, that's what's going on. Or I don't get this, right? So I won't use the term myogram. I mean, that's just a term for, right, whenever you see the word gram, it's a tracing of some sort. So graded muscle response, these are just myograms of twitches here. So it's giving you a compare and contrast. So this is the best way to think of, again, summation. Because you'll see that I don't let my muscle relax. Notice that? That's what I was talking about. It was very key in summation. Summation is I keep stimulating. All right? I won't use the word tropé, but that is the term we use for the appearance of this. It's a stair-stepping. Okay, so here again, just like on the last figure, it looks a little smaller, but same idea. Right? I did my stimulation, released my acetylcholine, right? All the events occurred that I said, right, to get us finally to get actin and myosin together. And tension started to build. And then I got to this point, and I started to relax it. But I didn't let it relax all the way. So not all the calcium went back. So there's calcium still out in the sarcoplasm at this point, and I send another action potential. And I get more tension. Because there was calcium already out, I'm releasing more calcium. More calcium means more opportunity for actin and myosin to bind, which means more tension. And there's clearly a point to this. That this isn't indefinite. You can't keep getting more and more and more tension. Right? But it does increase the force just by increasing the frequency or number of action potentials you send. Okay? This is really kind of your maximum here. And when you have repeated stimulation, right, see how this muscle is not relaxing? Right? This one was starting to, you just didn't let it go all the way until here. This one, you just kept sending action potentials. And you've probably been there. You're carrying something and you're staying in position. We call that tetanic or tetanus, sustained contractions, tetanic contractions. And they're just the frequency, right? So if you're carrying something for long distances, I always think of children. If you're a parent, you know this really well, right? You're carrying your child, you're staying in sustained contraction with that child with a load, your kid, right? And you're just gonna, you get as much force as you can, and you just don't relax. All right? So, now, the really good news, again, because like I said, we're simplifying this chapter simply because you're introduction. The really good news is that you have so many muscles, right? You have like 650 muscles in your body. All right? And they work as groups. You know, we probably, maybe that didn't get emphasized enough to you in lab because we were so busy naming each one, but they work in groups, right? So we didn't even go into the postural muscles back here. You just have so many groups of these, and they collectively, some of them we call the erector spinae because they're keeping you upright, especially with spine. And so what they do is they actually trade off through the day. So they achieve tetanus because obviously you're not moving, right? And you have to keep that force because gravity wants you to lie down. You know, so, you know, that's, gravity's always bringing us down, right? So you are overcoming that, and through the day, you just switch off. So one group of those muscles, or subgroup, would achieve tetanus, and they'd generate only the amount of force needed, and then they'd eventually start to fatigue, and then they'd switch off, right? And then somebody else would take over for a while, and so forth and so on. So your muscles really tend to work very coordinated as groups, but we're, you know, again, it's an introductory class, so. Muscle tone, right, is a constant tension, right, over long periods of time. So you do. And it's not until you die. Even when you sleep, you have some muscle tone. All right. So keeps us in positions. So depends on a small percentage of the motor units, as I just said, being stimulated, achieving tetanus, and switching off. All right. I'm not going to ask you a question on muscle tone either. I didn't write one. All right. I did write questions on this. All right. So types of muscle fibers. So this is a fun one. And I used to, in your Holes book, they talked about the twitch. And I tried to correct hopefully everything to oxidative and glycolytic. Because those are better terms. The twitch does refer to what we talked about, that some of them, these muscles, when we see fast and slow, well, they vary in those latent periods enough and how quickly they can contract. So when it says slow, they are, right, achieving contraction slower. And when it says fast, they are doing it faster, okay? So some books call these twitches, like fast twitch, slow twitch. They add, again, oxidative glycolytic to pay reference to the source of energy, right? If you're an oxidative, you're using oxygen effectively and you're doing aerobic respiration. If you are a glycolytic fibers, you prefer to do glycolysis. And remember, that works well with fast because breaking down glycogen is very fast in comparison to doing cell respiration, which has, not 10 steps like a glycolysis does, but 40. Okay? So it works together. And here what we do is we want to get you these idea of patterns. Now, let's be very clear. We are mixed in these. We're talking about the fibers themselves, right? So the muscle fibers, and then they're grouped together. So in a muscle like the biceps, that you guys, because you're looking at the big pictures, right, in lab, or, right, the gluteus maximus, or the sternocleidomastoid, or the orbicularis oculi, right? In those groups, sometimes there's a clear preference for one type versus the other, and sometimes they're just mixed. Yeah, Kim?
Have they proved whether or not you can change this?
There is suggestion that it can be, you can adapt it, but there's a genetic predisposition that you probably can't overcome entirely.
So it's limited to the variability you have.
Yeah, so, okay, because this is due to the myosin, ultimately, because myosin is the thing moving, and it's different types of myosin, which is in our genes. So, again, how fast you contract, how you produce ATP. That's really what we're looking at in these names. So I'm going to ask you a couple questions. I have like 2 or so that say, which of the following would be true of slow oxidative, which would be true of fast glycolytic, all right? Because the fast oxidative are intermediate, so they don't make a, you can write questions about that, but it's really, when you're comparing in introduction, slow and fast, slow oxidative to fast glycolytic is a really clean or clear comparison. So again, contraction is slower, not a lot, but it is slower. They are less likely to fatigue, use aerobic with oxygen, oxidative, more mitochondria, so you can definitely manipulate that. You can easily manipulate the number of mitochondria in a muscle. But again, that doesn't change it outright. More blood, yeah, because that's going to help me with that aerobic respiration, and myoglobin. So slow oxidative are your dark meats. Fast oxidative, again, this is an intermediate, so they are faster to contract, that's in the name. They still use aerobic and produce lots of ATP fast, but switch. These easily switch to an anaerobic, and they are more likely to fatigue than these guys. And they do not have myoglobin, right? And then fast glycolytic, these are going to be the quickest in that sense. They really prefer anaerobic glycolysis, which means they're not going to last long, right? They're going to be done for really quick movements. And so they get tired very quickly, again, because they're not efficient with their ATP. These muscles store lots of glycogen, very less mitochondria, no myoglobin. These are clearly white. So this is your light meat. And I say all you have to do is look at organisms and think about this. You look at chickens, where's the white meat on a chicken and where's the dark meat? So where's the white meat? It's on the pectorals, right? Right? Would that make sense, right?
Yeah.
And where's the dark meat?
The legs.
In the legs, yeah. Your thighs and your leg on a chicken because they run and run from predators. They don't fly. They plummet, but they don't fly. Hey, look at a duck or any migrating bird. Where's the dark meat? The slow oxidative. Where is it? If you're migrating, where are you going to have your efficient, less likely to fatigue, flying over an ocean?
Wings.
The pectorals, right? Because the wings are the structures, right, y'all? It's the movement. So those wings are never going to get very muscular. It's the pectorals that move them that you're going to want to really not tire out, right? And so if you look at ducks, their dark meat is really rich pectorals. And the legs wouldn't be nearly. Okay? So do think about that. Humans do have white, right? White, light, and dark meat. We do. But again, it's what you're going to look for, and what I'm going to ask you to think about is stuff like, well, if I do quick things like eye movements, that would clearly be a fast glycolytic. I'm not generating a lot of force, right? And here's my application, right? Chickens don't fly, ducks do. Slow twitch, fast twitch, again, those are the old terms, but there's no clear separation, again, with some exception, right? Postural muscles, again, your posture, you got to maintain that for a long time. Those are most likely to be more slow oxidative, okay? Frequent movements, that's going to be more of a fast, right? Again, fast oxidative in the lower limbs. And then fast glycolytic is like weightlifting, sprinting, okay? So I've kind of given you, and I do believe I have a question that does say, so which of the following? All right? Last part, and there's a couple questions on this on your test for sure, is comparing cardiac to skeletal to smooth, okay? So cardiac muscle is very similar to skeletal. And we have a whole chapter devoted to that in AP2, so we just want to remind you about the other muscle at the end of this chapter, right? Shorter, generally one nucleus. They're often branched and they have these intercalated discs. I'm not going to ask about those, but that's there. More mitochondria than skeletal. We already talked about that because you can't fatigue, right? Cardiac. Lots of myoglobin in cardiac muscle. They are autorhythmic, so they don't necessarily depend on the nervous system. There's localized changes in hormones that affect cardiac and smooth muscle. Right, so we've got gap junctions that let them depolarize. So again, I'm not going to ask questions about that. That's a very important aspect of them, but it doesn't lend itself well to questions. Gap junctions, desmosomes, so adaptations. Calcium is still really important, as is ATP, as I already mentioned, right? Sustains the depolarization, so there's a longer contraction. They call it the plateau, but again, here's an important point. A lot of the calcium, while cardiac muscle has a sarcoplasmic reticulum, a lot of the calcium they're dependent upon comes from the blood, which is why you needed parathyroid hormone. Because the calcium allows the contraction to be maintained, and it's during the contraction that you're pushing blood. And so you have to maintain the level of calcium outside the cells, which means your blood calcium, which is why parathyroid hormone's handy. All right, smooth muscle, not striated, right? So not organized as actin-myosin, spindle-shaped, one nucleus, so similar like cardiac in the nucleus, but not in the organization, right? There is actin-myosin, again, so they're just not organized in the same way. They kind of have this, again, very interesting arrangement that helps with the different, again, still generating force, still doing contraction. Calmodulins used instead of troponin. I didn't ask a question about calmodulin, but I will ask you about some basic differences. Calcium, there's a sarcoplasmic reticulum, but it's not really well defined. So smooth muscle, like cardiac, relies on extracellular, the outside calcium. Okay, so again, how do these things compare and contrast? So at least like two questions I had on the test, I'm pretty sure. Let's say how's it like, how's it different? It might be cardiac to skeletal, it might be cardiac to smooth. Again, you just create a table. It's really the easiest way to organize this information and say these things they have in common, these things are different. And if I've told you I'm not going to ask you about comments, I'm not going to, you should understand that actinomycin are not arranged the same. You don't have to know about dense bodies or the specific arrangement. Okay? And I just include too your erector pili that was in the skin, because you might be asked that on your lab practical, right? Because the lab practical does include skin. So that erector pili is also smooth muscle. And ciliary muscle that you're going to talk about in the eyes, that's also smooth muscle. All right. So it's contraction. Again, I'm not. Varicosities is a term for what it looks like. I'm not looking to ask that. It's what we're saying here is it's organized different. All right. So it doesn't, like cardiac, it doesn't absolutely require nervous stimulation. Skeletal does. Skeletal's not doing anything if it doesn't receive that acetylcholine. But cardiac and smooth have options. They have other ways to stimulate contraction. All right? And so they do have groups that we call pace setter cells. And again, not going to ask you a question on that. You do have to know these are involuntary though. You are not aware of the contraction. You're not telling it. All of your skeletal muscle, with the exception of reflexes, your skeletal muscle is being told by your right up here, the big cerebrum, and you're aware of that. You choose when you move and where you sit and what your position is. Cardiac and smooth do not. They do still use acetylcholine, and they do use norepinephrine, and we will talk more about those at the end of this course. Right? They also have stretch in the fibers, so physical changes in their length will cause them to contract. So lots of different things. You again need to understand that smooth is involuntary and does not solely rely on neural stimulation. All right, it does get organized, and I didn't ask questions about single units and multi-units either. All right, but this is just part of, again, part of my job is explaining what you are reading and what the rules are for these muscles. So we have differences in the way they respond. All right, and so it's kind of like I said, this just gives you a compare and contrast. There isn't really an opportunity in AMP2 for me to even bring this back up to students either, all right? That they, again, you've got a lot of them. I said more common. This is what we see in the hollow organs, the stomach, esophagus. I mean, your whole entire digestive tract has got smooth muscle all around it. Your blood vessels, with the exception of capillaries, have smooth muscle wrapped around them. You've got a lot of smooth muscle inside the body, all right? So here, this is the multi-unit you find in your blood vessels and respiratory. This is your digestive tract and other tracts. All right? So I'm going to skip this a second. Just remark that the regeneration of muscle proteins by satellite cells and pericytes, I do ask that question, and I didn't put it on here specifically, though I did ask that question in you guys' group questions. Where do I put those? And the idea that parasites, right? Not the A, and the SITE. These are a specific type of cell that smooth muscle have, which is why they can successfully regenerate to some extent. Okay, these are absent in your cardiac and your skeletal, right? They do retain satellite cells. In fact, if your instructor didn't point this out to you, there's one on this, right here. That's a satellite cell. It's found outside the muscle cell itself, right? Because there's a sarcolemma. But it's here to help repair this cell. Because even though we can't get the cell to divide, we can restructure part of it. So this is a satellite cell.
So it's absent in cardiac and skeletal?
So the parasite is. So the satellite cells are there. And they help with some repair, but the parasite is a cell that actually does a better job of reproducing the cells. And the satellite cell is there, right, as a basic repair of structure, but not getting the cells to divide. Okay, so that last part talks about that. All right, so I include this. This is really, I don't talk about levers. In the last, I included a little bit of this information just to reinforce what you guys are getting in lab. You should be very much aware of origins and assertions right now, and I do have a question on the test regarding. I define it and say, which is this? Okay. And the bottom line is the insertion is the one that does the movement. It moves towards the origin in most circumstances. All right? I did not ask anything about synergists, antagonists, or agonists. These are all terms when we talk about muscles and how they're grouped together, but the class and time doesn't lend itself well to really discussing that in detail. If you were not aware of this, I do have a couple questions on the test that talk, and hopefully, again, your lab instructor has made this very clear to you. The muscles are not randomly named, right? They have, if you look at their names, they have a number of them are shaped, like the trapezius, the deltoid, you have rhomboids, right? We're telling you what they look like when we give that name, all right? So that's why, you could guess that in knowing that. So, location, we got the pectoralis here, as I've already said, all right? You've got femoris, you've got brachii, right? Your oris and oculi, those are all locations in a name, aren't they? We're telling you where it is. Temporalis is a great one, right? Yeah, there it is. Origins and insertions, the only one we have you guys learn is the sternocleido, mastoid. All right? Numbers of origins, that's the biceps and triceps, right? You had biceps here, you have biceps here, you have triceps here, and there are other ones that are as well, but these are the ones we had you know. Right? There's a biceps femoris and there's a biceps brachii. So often we're mixing in the name, and that'll make your lives easier. Size, right? We've got the pectoralis majors and minors, right? We've got the vastus, right? Lateralis, medialis, intermedius, all big. And then where are they located? Function. There's a whole lot of flexors and extensors and pronators and supinators, but you really didn't have to know, right? Those, obviously, right? But there are a lot of them that are named for that. And then orientation of the fibers. So the classic ones, right, are here. Your abdominal ones, aren't they? Hands down, they are named for the direction that the fascicles are running. And the fibers will run the same way, right? You have a rectus, straight, right? And then it's abdominis for location, but rectus. You have a transversus. You have an external and an internal oblique. Every name is telling you what do the fibers, and therefore fascicles, look like. Because they're only going to shorten in the direction of the fascicles. So combinations of those will be part of your... So if you haven't embraced that, please do. You still have time certainly before my test, but your practical is definitely set in stone. You're doing it Wednesday or Thursday, unless, again, you arrange with your instructor. So I have a chapter 11 up there, but again, all I'm after is the same stuff you're learning in lab. So if you don't look at chapter 11, you don't look at chapter 7 and 8, you're going to be okay as long as you paid attention in lab, right? And